Questioning the genetic theory of cancer. If cancer really is some kind of monster, entirely foreign to the body and living inside us, then we're probably doing the right thing by trying to kill the cells during treatment. However, if cancer is not a murderous mutant cell, and these treatments are making the health of patients worse, then we need to know so that we can stop using them. It's time to question the genetic theory of cancer to find out for certain. In search of another paradigm that could adequately explain the underlying cause of cancer and why the war on cancer has been such a failure, I stumbled upon a fascinating series of studies that completely contradicted the genetic theory of cancer. If cancer were a disease of genetic origin, then none of the following observations would have occurred. Clone mice from tumor cell DNA. In 2003, a group of scientists from St. Jude Children's Hospital in Memphis, Tennessee, took cells from mouse brain tumors and then cloned a mouse using the DNA contained within that tumor cell to see if the cloned mouse would harbor cancer or be all messed up. The results? The study was published in the journal Cancer Research, and what they found was that the cloned brain cancer cells directed normal development in the mice, both pre and post implantation. So they took mutant DNA from a mouse brain tumor cell, cloned another mouse from it, and that mutant DNA directed normal development in those cells. That completely contradicts the somatic mutations theory of cancer. Frog egg tumor transplants. This is the next study. In 1969, a group of researchers injected tumor cells from tadpoles into frog eggs to find out the condition of the tadpoles that would emerge from those frog eggs. Of course, after the tumor cells were transplanted, the eggs contained the mutant cancer cell DNA. So the question is, were the tadpoles that emerged cancer-ridden or not? If the somatic mutations theory is correct, they would have to be. The results of the study were as follows. From within the eggs emerged healthy swimming tadpoles, demonstrating once again that mutated cancer DNA can direct normal development. Harry Rubin, Professor Emeritus of Cell and Developmental Biology from the University of California. Look at this guy. If you're watching the video, you can see him on the screen just smiling away. This is what happens when you live a life of adventure and scientific discovery. Anyway, Harry Rubin demonstrated in 2006 that cells can have hundreds of mutations and still behave normally within the organism. The study reports, and I quote, "The use of a reporter gene in transgenic mice indicates that there are many local mutations and large genomic rearrangements per somatic cell that accumulate with age at different rates per organ," and here's the key, "and without visible side effects." You can have hundreds of mutations in your cells and there'll be no symptoms from that. The cell function will be normal. Not only does this fly in the face of the somatic mutation theory of cancer, this flies in the face of the popular dogmatic idea that the genetics of a cell are important at all for cellular function. They don't seem to direct cell behavior whatsoever if you can have hundreds that are mutated and the cell still functions normally. And one more fascinating study. Cell cytoplasm-swapped CYBRIDS. What is a CYBRID? Since the 1970s, scientists have been experimenting with swapping normal cell cytoplasms. So if you aren't aware, a cytoplasm is a part of the cell which contains the energy-producing mitochondria, not the DNA. And the study was about taking the cytoplasm from a cancer cell and putting that into a healthy cell and seeing what happens. And conversely, taking the cytoplasm from a cancer cell and putting that in a healthy cell and seeing what happens to that healthy cell. Anyway, the resultant cell that has the cytoplasm swapped is called a CYBRID, which stands for cytoplasm hybrid. So when scientists transplanted normal cell cytoplasms into cancer cells, so a cancer cell obviously has the mutated genetics, and you put a normal healthy cell cytoplasm containing the mitochondria in that tumor cell, what happened to it? Well, in this study, the cancer cells transformed back into normal, healthy, functioning cells. So not only does that fly in the face of the somatic mutations theory, that the genetics have nothing to do with a cancer cell or a cell becoming cancerous, but it also suggests that a cancer cell is completely reversible without the need to attack it in any way with knives, poisons, or radiation. You can turn a cancer cell back to a normal cell by putting a healthy cell mitochondria within that cell. And when cancer cell cytoplasms were conversely transplanted into normal cells with normal DNA, yet cancerous mitochondria, the cells turned into cancer cells. So what these findings show is that mutant cancer cell DNA does not cause cancer, and that it's the mitochondria that appears to dictate carcinogenesis.
Hey, it's Mark. Thank you for watching my video. If you enjoyed it, give it a thumbs up, and definitely hit subscribe so you don't miss out on future videos when they come out. Also, be sure to pick up my 3 free ebooks, one on chemotherapy, the other on sodium bicarbonate for cancer and other diseases, and a red light therapy dose guide by clicking the link in the description below. Last but not least, check out one more video on my channel right here, and I will see you in the next one.